Hi, this is our Lac Operon project by Franklin and Daniel Martinez. So the Lac Operon is designed by E. coli to break down lactose. They need to be able to do this because when they live in a host that takes in lactose, they need to be able to break it down so they can capture the energy for themselves as well. And the DNA of the bacteria includes this. So this is the promoter, the operator, and the genes. And then the repressor holds back the operator it holds back the DNA polymerase from making the genes constantly, since it blocks the DNA polymerase's path as it tries to go through. So the promoter is the region of the DNA where the RNA polymerase can grab onto, and the operator is the on and off switch for the gene making, and the repressor is attached to this to prevent this when not needed. So when the bacteria receives lactose, this is lactose, and there's actually lactose in there for it to break down now. The lactose attaches to the repressor, and after this happens, the repressor changes its shape, it goes away, and it releases from the operator. And after this happens, the RNA polymerase can now go through, since there's no longer a repressor holding it back, and it goes through the genes, and now it can transcribe and translate a protein and make a protein for breaking down the lactose. So it makes this protein after the translation process is over. And this protein is called a lactase, and it can break down lactose into simpler sugars. So it goes in here on the lactose, and then it breaks it down into two simpler sugars, so glucose and galactose. And it does this for every single lactose there is. And after every lactose is gone, the repressor can now attach back to the operator, since there's no longer a lactose, and there's no reason for it to make the lactate anymore, which then goes away, since there's no longer any lactose for it to break down. And now the RNA polymerase can no longer continue to go back to the three genes, the lac Z, Y, and A, that codes for the lactate to break down lactose, since the bacteria no longer needs to do that. And that's the Lac Operon phase. And since it's a cycle, if there's more lactose, it continues and it does the same thing over again, and the lactose attaches back to it, and then it does the same thing over till the lactose is gone, and then it stops making, and then it continues and it stops. And that's how the operon works. Thank you.